today we're going to discuss bacterial metabolic behavior, and this is a very important quality that bacteria have that are very different from other organisms. And we're going to examine how, for instance, bacteria can live in environments that have no oxygen at all and certainly thrive, and other bacteria can certainly live in environments that have oxygen, and some bacteria actually can go back and forth between living in environments that have oxygen and environments that don't. So to give us a review, energy metabolism is really how do we get energy from the food that we eat. So all energy, at least as far as organisms that engage in aerobic respiration, comes from chemical energy, from food, from plants, from animals. And because we're organisms that can eat actually both plants and animals, our food is composed of both. And through various biochemical mechanisms, we utilize these foods, we break them down, and we produce ATP, which is the energy currency form that energy is really stored and created in all organisms. And then by metabolizing ATP, we produce waste products, carbon dioxide, and water. And then the energy that we get enables us to move, to engage in biochemical processes, and then of course we give off heat. So in terms of bacteria, that this metabolic behavior is a very important criteria for classification. So we'll see that there are some, as I was mentioning, some organisms that are anaerobic, some bacteria that are aerobic, and some that go back and forth. So to review again, that autotrophs are organisms that can produce their own energy without having to consume another organism. And they're, for instance, the plants utilize the sun and also carbon dioxide, that carbon dioxide is the source of carbon, and then they produce glucose from the sun and carbon dioxide. Most of them do this through the process of photosynthesis, and they obtain their energy directly from the sun. Now, heterotrophs, on the other hand, they are organisms that are required to consume other organisms in order to stay alive. So they don't use carbon dioxide, rather they rely on oxygen, and they obtain carbon from the environment through other organisms. And in fact, that's how they survive, is they have to eat other organisms. So even herbivores or vegetarians, they certainly eat organisms as well. And constantly, in terms of the earth, for instance, there's this constant recycling back and forth between autotrophs and heterotrophs. So to see it in a pictorial form, you can see here that the autotrophs use the sun plus carbon dioxide, and they create glucose, and the heterotrophs consume the plants, and then some heterotrophs consume other organisms, and they require oxygen, and they develop or they derive their glucose from breaking down more complex forms of glucose, and then they give off carbon dioxide, which is then taken up by the plants with the sun to create glucose. So we have this constant cycling back and forth between creating glucose and then breaking it down. So metabolism really is the sum total of all biochemical reactions that occur in the body or in a cell or even in another organism, of course. And really that this metabolism is directed and regulated and occurs through a series of enzyme catalyzed reactions. So these reactions are essentially regulated and initiated and terminated even by reliance on enzymes. Catabolism, again, to review, is the process by which we take more complex molecules and we break them down into simpler forms. And by doing that, we produce energy. Anabolism is the opposite. Anabolism is the synthesis or creation of more complex molecules from simpler ones, and that by definition requires energy. So these processes, these metabolic processes, go back and forth. Sometimes they're occurring simultaneously. Like for instance, the human, we consume food, we break it down, we use those simpler products, and then we synthesize them into more complex structures in the body and so forth. So even within one organism, we go back and forth, and then certainly in the animal and plant kingdom, we do as well. So the definition of cellular respiration now is to create or produce ATP, which again is the currency molecule of living beings or of life, and creation of ATP. So we go from ADP, we add a phosphate, and then we get ATP. So we get 3 phosphates. We start with 2, we add on, and now we have 3, and then we can certainly go in the other direction. So depending on what direction we're going in will tell us whether or not we have to add energy or we get energy produced or liberated, so to speak. So this ATP is constantly going back and forth depending on what cell or what process or what organism or what conditions. And so the organisms all engage in this process of going back and forth between ADP and ATP. So aerobic respiration is the production of ATP in oxygenated environments. By definition, oxygen is required for this process to occur. And the final electron acceptor is oxygen itself. Now, anaerobic respiration is the production of ATP in environments where oxygen does not exist. And these anaerobic bacteria thrive in these environments without oxygen, and in fact, if you add oxygen to their environment, they actually die. So the electron acceptor in that particular situation is not oxygen. So glucose, again, is the energy currency molecule of life, and it's the currency molecule for both heterotrophs and autotrophs. Glucose has 3 major outcomes. It can be stored, it can be oxidized. Now remember, what does oxidize mean? It means broken down. It can be broken down through glycolysis, and it can be broken down through this pentose phosphate pathway, and I have a picture on the next slide. The pentose phosphate pathway is the pathway by which we produce sugars that get combined in nucleic acids. And more commonly, we oxidize glucose into pyruvate. Now, further from this place, from pyruvate, is where we have anaerobic or aerobic environment. So this is with oxygen, and this is without oxygen. And both of those processes start with pyruvate. So this first process really is the same for both anaerobic and aerobic bacteria. So here we go again. So here is a more refined picture of what I just showed you. So we have our glucose. It gets broken down into pyruvate, and then it can either go through the aerobic pathway and turn into acetyl coenzyme A, which then goes down the citric acid cycle to oxidative phosphorylation. Or we can go anaerobically through the lactate pathway, or anaerobically through the ethanol fermentation pathway. Now, the word fermentation really is a general term that indicates that the process is anaerobic. However, fermentation is often also thought of in terms of the classic ethanol fermentation and not the lactic acid fermentation. And we're going to see that this lactic acid pathway isn't just employed by bacteria. In fact, humans engage in this anaerobic respiration as well, and that is exemplified by a sprinter, for instance, who runs a 100-yard dash in, I guess Usain Bolt did it, I think, in 9 seconds. And a short sprint like that is generally almost completely anaerobic. The same idea is when we dive and go underwater and swim underwater, we're not able to derive oxygen from the water, and yet we're able to contract muscle and engage in energy. That is all anaerobic. However, we cannot obviously stay underwater for all that long of a time. So the anaerobic pathway in humans is very short, but it does certainly exist. And then, of course, it's prominent in these bacteria who live in environments that don't have any oxygen at all, and they actually engage in the very same process. So in terms of the aerobic pathway, we have 3 stages. We have glycolysis, we have the citric acid cycle, and we have oxidative phosphorylation. And just on a general scheme or a generalization, we take glucose, we add oxygen, we get carbon dioxide and water as waste products. So that's exactly what happens in aerobic respiration on a much simpler level. Now, to get a little more complex, each of these stages have many biochemical reactions that both prepare the molecules to be broken down, and then ultimately they get broken down. So in terms of the first stage, the first stage is production of acetyl coenzyme A, and that stage is called glycolysis. And all anaerobic and aerobic bacteria, and aerobic humans for that matter, they all engage in the same process of glycolysis. So after the acetyl coenzyme A production occurs, and you can see here it isn't just glucose, we can also have amino acids and fatty acids, and they actually go straight to acetyl coenzyme A. They don't go through the pyruvate path, which is exclusive to carbohydrates. So once we get to the acetyl coenzyme A level, then we go down the citric acid cycle, and then ultimately we engage in oxidative phosphorylation. So you can see here that the ultimate outcome is production of quite a lot of ATP, that this process is very efficient at producing ATP. Now, in terms of comparing respiration, which is a process that requires oxygen, comparing that to fermentation, which again is another word that means anaerobic respiration, you can see that here we go, glycolysis, pyruvic acid, we go through we get to acetyl coenzyme A, we go through the Krebs or the citric acid cycle, and then we come down to the oxidative phosphorylation. And you can see that here we have production of ATP in the citric acid cycle, as well as production of ATP in oxidative phosphorylation. Now, in fermentation, on the other hand, we start with pyruvic acid, just like with aerobic respiration, and we go through these various steps of fermentation, and we come out with these end products, and we come out with some ATP, but the ATP from fermentation is considerably less than the ATP that comes from respiration. So to compare eukaryotes from prokaryotes, now remember, prokaryotes do not have organelles, and they do not have, for instance, mitochondria, whereas eukaryotes do. So the glycolysis part is the same for both types of organisms. The intermediate step is similar or the same for both organisms. Now, the eukaryotes have the Krebs or the citric acid cycle and electron transport chain or oxidative phosphorylation. Those two processes occur in the mitochondria, but because prokaryotes don't have one, they continue to engage in the Krebs cycle in the cytoplasm, and then they use the plasma membrane just in the same way that the mitochondrial membrane is used in terms of the electron transport chain. So anaerobically now, remember one of the pathways, the anaerobic pathway, produces lactic acid. So this is the production of lactic acid from pyruvate to lactate, and it occurs through this enzyme called lactate dehydrogenase. And you can see that it facilitates the addition of hydrogen ions that break apart the double bond here, the carbon-oxygen double bond, and adds the hydrogens to break up that bond. And this is the amount of energy that's produced. So again, there are two types of fermentation. There is the lactic acid fermentation, which, remember, humans go through that as well. And then alcohol fermentation, which is the classic production of alcohol through putting in, certainly allowing fruits to spoil, for instance. We get alcohol that way. And that certainly occurs without oxygen. And lactic acid fermentation also occurs without oxygen. So we do get ATP. It's just not as much as the ATP produced under aerobic respiration. So in particular, alcohol fermentation, which again is one of the types of fermentation, is considered kind of generally as any spoilage of food that occurs from microorganisms. Alcohol fermentation also is a process that produces alcoholic beverages, and generally that's done through the use of yeast. And then it's also considered as any large-scale microbial fermentation that occurs without oxygen. And there are some industrial processes, for instance, that rely on this particular respiration method. So to continue with alcohol fermentation, so that yeast and other microorganisms ferment glucose to form ethanol and carbon dioxide, and that is rather than the lactate that occurs in the other type of fermentation. But we still rely on that glycolysis pathway is still the same. So we start out with glycolysis, okay, and we get pyruvate, and then pyruvate is reduced in a different way than it is in the aerobic pathway. It's reduced and catalyzed by different enzymes, and CO2 comes off the molecule, and then it's further reduced to ethanol with another enzyme, alcohol dehydrogenase. So the lactic acid pathway, on the other hand, is considered the classic anaerobic pathway. Excuse me. Some bacteria exclusively rely on this type of fermentation to survive. It doesn't require oxygen. We don't have the Krebs or the citric acid cycle or electron transport chain, and we have an organic molecule as the final acceptor. So here's a nice flow chart of all these different types of fermentation. Here is lactic acid production. Here, excuse me, are the other forms of fermentation. And then here's a nice chart. You can see that these different end products are certainly produced by these various methods and different microorganisms. So the traditional alcohol production is often done by these yeast organisms. And then these various dairy products, cheese, is also produced by lactobacillus, which is a bacteria, of course, and certain breads and sauerkraut and sausages, those are all produced by bacteria. Excuse me. And, you know, we have other certainly industrial products that occur with yeast and other sorts of fungi and also bacteria. And these are, you know, important industrial chemicals that are certainly produced through utilization of these microorganisms and, you know, relying certainly exclusively on anaerobic respiration. So the first example, or the first question, asks us, why do you think food spoils? You know, what happens? What causes food to be spoiled? Now remember, our first lecture, there's no such thing as spontaneous generation. Okay, so we can't just spontaneously have organisms show up in our food. So there are various ways and reasons. So certainly the air, right? The air in the container can have microbes. The food can have microbes. And both of those, by definition, pretty much have to function anaerobically for that food to spoil. Now, especially if you've got a really tight container, right? So remember that one slide talked about alcohol fermentation as including the food spoilage. So basically the answer to that question is it occurs that spoilage occurs through alcohol fermentation. So which pathway is the most efficient pathway to produce ATP? Number one, by a long shot, is the anaerobic, or excuse me, anaerobic, the aerobic. The aerobic pathway by far produces a lot more ATP. In fact, it's usually about 30 to 32 molecules. Here we go, sorry about that. Compared to maybe 2 to 4 with anaerobic. So really, if you're trying to develop a particular method for creating ATP, doing it in an oxygenated environment is the most efficient method for creating ATP. And anaerobic organisms do just fine with the ATP that they produce. So certainly if an organism has always been anaerobic, it's never going to know that it could have a lot more ATP, but certainly those organisms do just fine in an oxygen-deprived environment. So the third question has to ask, again, what is fermentation? So remember, fermentation is another word for anaerobic respiration. Okay, and that, however, usually classic fermentation is really the production of ethanol, whereas the actual anaerobic classic pathway is the production of lactic acid. But both of them really are generally called fermentation. So that concludes our discussion about bacterial metabolic behavior. Thank you so much for visiting educator.com.